really much further ado, I will hand over to Dr. David Roberts, who's going to give tonight's talk, which is on, as you can see, the curious case of the man, the lady, and the monkey. So looking at monkey orchids and explaining some of the conservation efforts that are going towards those. David is from the University of Kent, and I will hand over to him.
Thank you very much. And thank you very much for inviting me to give a talk this evening. So yes, my name's Dave Roberts. I'm at the University of Kent in the Durrell Institute of Conservation and Ecology. Prior to moving here about 15 years ago, I worked at the Royal Botanic Gardens, Kew, as an orchid specialist in the orchid herbarium. So in terms of my background, and so I did my PhD on orchid and orchid reproduction. And so this talk is kind of going back to, in part, my PhD days, purely from the point of view that I worked on pollination and reproductive biology of orchids. And during lockdown, I got asked whether or not I'd be interested in working at a particular site. And so I managed to kind of restart my orchid pollination ecology work again after a long time, 25-year hiatus in working in this area, or 20-year maybe. Yes, tonight I'll be talking about man orchids, lady orchids, and in particular monkey orchids. And the reason why monkey orchids are particularly of particular importance is that they're extremely rare. It's one of the rarest plant species in the UK. And so the monkey orchid, or Orchis simia, is one of 5 species of orchis found in the UK. As I say, it's one of the rarest. The military orchid is also very rare. But of those 5 orchids, 4 of them are what we call anthropomorphic, in that they look like things that look like humans. In the case of the man orchid, as you see on the left-hand side, and then we have the lady orchid. This one seems to have culottes on, but often it has a dress. So the labellum at the bottom looks very much like a dress. You have the monkey orchid, which is quite thin labellum, so lower lip, with long lobes, very thin, long lobes, and a small appendage in between in terms of the tail. And then finally, you have the military orchid, which you can see here. And this has is a bit like the monkey orchid, a bit more robust in terms of the flower, but has a much more definite kind of helmet in terms of where the military orchid comes from. So the sepals and lateral petals come together to form this kind of helmet structure, compared to the monkey orchid, which does form a structure like that, but it's can be not as distinct in terms of kind of the kind of density that they come together to form this helmet. So those are the 4 anthropomorphic orchis species in the UK. But of course, there is the early purple orchid, Orchis mascula, which is a relatively common species as well. So in terms of the species, Orchis simia, it's extremely rare, as I mentioned. It's a Schedule 8 species, therefore, to do any work on it requires a license. So even if I want to do pollination studies, removing pollinia and things like that, I would need a Schedule 8 license. Now, its rarity is down to the fact that it's only found at a few sites. The 3 main sites are Hartslock in Gloucestershire, and then 2 in Kent. One is a site near Faversham, and the other one is Parkgate Down, where the seed originated actually from the Faversham site. Occasionally it pops up in other locations. It seems rather ephemeral in those. but these are the three main sites that when we think about Orchis simia in the UK, these are the ones that we automatically think about. Now, as with other orchids, because orchids in the main, there are a few tropical orchids in the main don't have chemical inhibition systems to prevent cross-fertilization. Hybrids do occur. Generally, orchids position the pollinia on different parts of an insect to prevent cross-pollination, but in some cases, this isn't quite as as good a tactic as say the chemical systems, and therefore hybridization can occur. And so with the anthropomorphic orchids, hybridization has been recorded, and for Orchis simia, it's well known at one particular site. Now, Orchis purpurea is found at all three sites in varying numbers, and also Anthropophora, the man orchid, is also found at the Faversham site, which is the site that I mainly work at. So there is the potential for hybridization to occur. At Harkslock, there is a hybrid swarm between purpurea and simia, and this is very well recorded, and there has been quite a bit of research into this by Richard Bateman and more recently by others at Kew. And then there is the hybrid between the man orchid and the monkey orchid. And this occurred in 1985. It was the account was written up by Richard Bateman. And it was a single plant that was found at the Faversham site. Now, it occurred at the Faversham site in 1985 and then disappeared for a while and then reappeared in 2016. And it's always been curious in terms of how this has occurred. Is it a natural event or is it a man-made event? And so this is what kind of resulted in my interest, is the extent to which hybridization between co-occurring Orchis species is a threat to the persistence of Orchis simia. And as you see in this great image from one of Richard Bateman's papers, you have at the top, you have various Purpurea simia hybrids from Hartslock. And then at the bottom, you can see, in bottom center, you can see the hybrid between simia and Anthropophora. And so you can see how they kind of integrate into each other. And so, as I say, this is a threat, and you can, you know, we do run the risk of losing Orchis simia to the UK, depending on the extent of hybridization that occurs. So Orchis simia, the Faversham site is a relatively long-term site. It's had Orchis simia there since 1955. And so it's historically very important. It was the seed source for the seeds at Parkgate Down, which is one of the Kent Wildlife Trust's sites. And so you can go and see them very easily at Parkgate Down during the season. And the other benefit of the Faversham site is that, unlike Hartslock, it has limited hybridization having occurred. As I say, there's only the case of the man orchid and hybridization. And again, this was a single plant that came up and then disappeared for a number of years and then reappeared again, but again hasn't been seen. So although Anthropophora has hybridized with simia at the Faversham site, and purpurea does occur at the Faversham site. A purpurea simia hybrid hasn't been recorded there at present. So my interest in this was looking at the extent to which hybridisation is a potential risk to the persistence of the anthropomorphic species, and in particular simia, and through looking at pollination ecology. Now, in terms of orchids, besides looking at taxonomy, a lot of work has gone into looking at pollination ecology. Of course, they're very famous examples in botany are based on orchids. Darwin, of course, wrote his famous book after writing The Origin of the Species, and of course, work on mycorrhiza. However, it's interesting that actually relatively little work has been undertaken looking at the pollination ecology of orchis, and particularly in the UK. So we don't really have a good idea of the extent to which there's overlap between potential pollinators to plants, and therefore the extent that this is a potential risk. So I started work at the site in 2022. I'm very grateful to the landowner who contacted me and asked if I would be interested in working on the population there. And so in the first year, I looked at seven, I monitored seven anthropophora, man orchids at the site, 22 purpurea, and then 100 Orchis simia, the monkey orchid. So you can see that it's a very large monkey orchid population compared to purpurea and anthropophora. So anthropophora at the site is generally limited to the grassland area of the site, with a single individual found in the woodland in association with Orchis purpurea. Orchis purpurea at the site is found behind a bank of conifers that separate it from the grassland area. It's kind of confined to that area, except 2 or 3 flowering plants that occur on the other side of this kind of conifer boundary, and therefore are close to the fence of the grassland area. And then Orchis simia is found across the site. A few individuals have extended into the woodland, but not in the area where purpurea occurs. So they've extended into that area, but predominantly it's found in, it's kind of very concentrated in a scrub area under a canopy. This scrub area has been kind of actively managed to reduce it being overtaken by other woody species at the site. But as I say, it's generally localized to that area. However, again, it is expanding across the site again. Talking to Richard Bateman, he was interested in terms of looking at where it occurs now and where it occurred back in 1985 when he went looking at the hybrid at the site. So this just gives you a kind of pictorial representation of the site. Purpurea in the purple, as I say, is generally found behind a conifer boundary. It's quite dense, so movement of potential pollinators between there is going to be tricky. There is a single man orchid found there. And then in the more open grassland area, you tend to find anthropophora, the man orchid, to the edge of the site and along the bottom. You then find this Orchis simia. You find it across the site up into the woodland, but it's a lot more sparsely distributed compared to where you find it under the canopy. It's not a dense canopy of trees on the opposite side of the site. So I went to the site during much of its flowering season, or at least just before the peak, and then through the tail of the flowering season in the first year and in subsequent years. So in this first year in 2022, I spent just under 52 hours sat looking at Orchis species. Spent a long time just staring at flowers, willing a pollinator to come along. I looked at all three species of orchis at the site and stared at them for nearly 52 hours. And during that period, I never saw an insect turn up with either pollinia on the head or any other part of their body, or see a visitor remove pollinia. So I spent a long time just staring at the plants, and I've done this in subsequent years. And again, never seen pollinia removed or deposited. However, I did record 52 visitors, so 52 insects that landed on the inflorescence and did search around the flowers, either during these 52 hours or through ad hoc observation. So while I was going round, either counting the number of flowers on a plant to look at how many were open and so on. So in terms of the distribution, of course, I concentrated mainly on simia to understand the pollination ecology of that particular plant. But you see that we had 23 visitors to Orchis simia, 6 to anthropomorphic, and 12 to purpurea. And this equated to, in terms of the inflorescence hours, so in some cases, I managed to look, there were 2 inflorescences next to each other. And so I managed to kind of stare at those willingly for an hour. And so that would count as 2 inflorescence hours. So Orchis simia, we saw on average, it was just over 3 visitors per inflorescence hour. Anthropomorphic was about half a visitor, so 1 visitor every 2 inflorescence hours. And purpurea had 0.38 visitors per hour, inflorescence hour, so that's round about 1 visitor every 3 hours. So in terms of the species that turned up, simia, we found 14 species on simia visiting, 6 on anthropomorphic, and 8 on purpurea. In terms of the Orchis species and the visitor groups, when you classified them into, say, flies and butterflies and bees and so on, there was no significant difference statistically between the different orchids in terms of the visitors. But the interesting thing that we found was hoverflies were dominant in terms of visitation. So Orchis simia, 2/3 of the visitors were hoverflies. Purpurea had even more, 70%, and anthropomorphic had just over 50% visitors being hoverflies. Now, hoverflies, it was interesting to see that it was hoverflies and see the impact between 2022 compared to 2023. Hoverflies like to lay their eggs in hollows in trees or other kind of pooling water areas. They produce what they call rat-tailed maggots. So they're maggots with this long tube that goes up to the surface to allow them to breathe. And so if you remember, of course, 2023, that summer was extremely hot. Plus, we'd already had a cold, wet summer, spring. And so I think this had a major effect on the pollinators. Sorry, 2022 had the really hot summer, which meant that when they laid the eggs, a lot of the pools potentially dried out. So in 2023, when we had the cold, wet spring, it probably had a significant impact on pollinators in that year. But as I say, interestingly, hoverflies are dominant. These are just some of the species that we found. Just to give you an idea of the diversity and also an idea of my inability to take clear, focused pictures of insects. I'm not the best photographer when it comes to this. So as you can see, there's a variety of different hoverfly species. Identifying them to species is extremely difficult in some cases because you generally need the males and you need to dissect out the genitalia to identify them. So some of them were identified to genus, but not beyond that. You can see there's a number of different bees found on simia, as well as the brimstone butterfly was also found on it. This is the man orchid, again, a number of different hoverfly species, but also other fly species and sawflies being found on the inflorescence. And finally, Orchis purpurea. Again, a number of different bee species found on there, a lot of different hoverflies, as well as this scorpion fly as well being found on it. So in terms of looking at the pollination ecology, what we're interested in is not just the species of insects that turn up, but the kind of overlap in the phenology between the three species. And so what we found, as you see, I started slightly late in May. Unfortunately, teaching got the better of me getting to the site earlier, but I started on the 13th of May in 2022. I got there a bit earlier in 2023. But you can see in terms of the distribution, purpurea seems to flower slightly earlier than simia, and then anthropomorphic flowers slightly later, but it's relatively rare at the site, and so kind of sits around the bottom, as you can see from the graph. Now, when you compare between the 2 years, you can see a difference in terms of the overlap of flowering. So in terms of the number of flowers that are open at any one time. As you can see, between 2022, you did see a shift with Orchis anthropomorphic having more flowers open earlier. Again, there was a number of purpurea, but less than previous years. And then you had simia, of course, dominating at the site. So you can see that there is a shift in phenology between the species. They're not responding the same. They're not moving earlier or later necessarily at the same rate. So one of the important aspects is understanding the fruiting success, so the number of capsules that are produced by the orchids, which gives you an idea of reproduction at the site, the extent to which it's occurring. And as you can see, Orchis simia is extremely low, less than 4% fruiting success. So only less than 4% of any flowers produce a fruit. when you start looking at anthropophora, again, this you then start seeing 20%, 30% fruiting success, and purpurea sitting in between at round about 10 to 20% fruiting success. So you can see that there is a very very much a difference between fruiting success between these different species, with simia having relatively low fruiting success. Now, all these species are thought to be deceptive in their pollination, that they don't produce a reward. Um, a reward, say, in the form of nectar. So in this case, they may have a strategy of just looking like a nice looking flower within the environment that attracts a pollinator. They collect, they go there, can't find any nectar. However, in the process, they pick up the pollinia. They may try another plant, another individual, again, not having any pollinia, sorry, not having any nectar, and then fly off. And this, of course, increases outcrossing. It is thought to be a strategy related to outcrossing. However, of course, the low pollination success may be of concern for Orchis simia. Now, there has been suggestions that actually these orchids may produce some nectar in the flower, in the depression around the entrance to um, the spur. And that may fit with the visitation of the hoverflies with particular mouth parts for extracting sugary solutions, as well as other fly species. So they may be trying to attract these sorts of species that may produce possibly a reward, or at least a kind of garner interest by the visitor to stay around slightly longer. Now, of course, I've mentioned Orchis simia has very low fruiting success. However, it's interesting that the fruiting success isn't evenly distributed. The heart of the population, underneath the shrub cover, the canopy to the side of the site, has very low fruiting success, about 2 to 2.5%. Whereas in the grassland areas, it has between 5 and 7%. So it may be that, again, as I've mentioned, they're using visual cues to attract the pollinator. If they are using deception, they're using a strategy of looking like a generic mimic within the site, that they're just look like a kind of a beautiful looking flower that attracts an insect to visit. So so they may be going for this sort of strategy, and therefore controlling of the shrubs on the site may be beneficial to allow insects to actually see the simia, Orchis simia, and allow pollination to occur. Now, one of the other aspect is also density. Because you have an area that is highly dense in Orchis simia, but they don't produce a reward, there is no incentive to maintain the insect to stay around that area. And so often what you find in deceptive orchids is that they'll visit the flower, as I mentioned, find that there's no reward, and then they'll do a long distance movement out of what is a poor resource area for them. And so maybe the high density of flowers in that area is attracting some pollinators, but because it's producing very little rewards there, the insect quickly learns and moves out of that area, and therefore results in low fruiting success. But of course, the question is, how many, how much fruiting success is required to maintain the species at the site, and that, of course, is a difficult question to answer. Of course, orchids produce lots of seeds, and it only, of course, requires a few of those seeds to germinate to result in the next generation. And so it would be interesting to look at the kind of threshold in terms of pollination success. The other aspect in terms of orchids is that while they may be pollinator limited, in that not all the flowers get pollinated, so they're pollinated, they're limited by the number of pollinators, they can also be resource limited, in that having fruiting success in one year results in such a strain on the plant that it dies down and doesn't come back for another year or two, or even more. And this has been recorded in some orchids. So even if it does come back, it may have a smaller inflorescence because of the energy that's been required to produce that fruit. So there are various playoffs occurring in relation to fruiting success. Just as an aside, you do see with simia that fruiting success and inflorescence size is correlated. So the larger the inflorescence, the more likely they are to produce a fruit. And this is seen in anthropomorphic, but not in purpurea. One of the interesting observations, just as an aside, that I found is that with anthropomorphic and purpurea, like many species, the flowers open from the bottom sequentially all the way to the top. Some at the top don't open, and they sometimes don't go all the way to the top in terms of opening of the flowers. But with simia, it seems quite bizarre. It seems to open from the bottom, from the top, and also from the middle. So there doesn't seem to be a particular direction in which the flowers open. Often they open from the bottom to the top, and the insect, if it's a rewarding species, at least the insect will, of course, follow the flowers up in a spiral motion, working their way up the flowers. And so simia it may be another strategy in terms of getting the insect to fly off, because essentially as the insect moves along, it gets to a flower that isn't open and therefore has a potential incentive to leave. And so this may be another strategy by simia to increase outcrossing. So one of the things I was interested in is the overlap between different species, the different species on the site. So comparing simia and anthropomorphic and purpurea. And in terms of the phenology, as you can see from the graph, but also statistically, purpurea has a greater overlap in terms of phenology with simia than does anthropomorphic. But as I mentioned, you do see this shift. As I mentioned before, you see this shift in terms of phenology. And so this relationship between phenology may not be the same year on year. But when we look at overlap between the pollinators, if we look at the visitors, I say pollinators, visitors because we haven't seen pollen removed or deposited. So in terms of the visitors, if we look at all the species that have visited the flowers that have been recorded, now you see a shift towards anthropomorphic having a greater overlap in the visitors compared to purpurea. So anthropomorphic has a greater overlap between visitors, but purpurea has a greater overlap in terms of phenology, at least in 2022. And this overlap in terms of visitors, again, is seen if we lump all the visitors together into different groups in terms of hoverflies and butterflies and bees and so on. You see this pattern again with anthropomorphic having a greater overlap with simia than purpurea. Now, one of the other observations that I had at the site were pseudopeloric flowers. I estimate out of the 100 flowers I looked at, probably 10 had what I think are were pseudo peloric flowers, in that the labellum, so with its kind of long arms and long legs and its tail, these were petaloid. So they were like the normal kind of ovate lateral petals. And so as a result, because you haven't got this long labellum lip hanging down with all its appendages, what you tended to find in these was that the flowers remained closed because the petaloid labellum was curling up like the other petals, the two lateral petals. And so the flowers were relatively closed. None of them had any fruiting success. And so it's interesting to understand how these are persisting at this site. Similar observations have been had in Kent in relation to Orchis mascula. I think I saw on Twitter, I can't bring myself to call it X, sorry. On Twitter, I saw there was a nice picture of Orchis mascula that was a pseudopeloric mutation there as well, where the labellum had been replaced by a petal-like structure. So in terms of discussion, so at the Faversham site, the hybrid between Anthropomorphic and simia was recorded in 1985. And there had been suggestions that this may have been due to hand pollination. How you get the two species accidentally mixed up, I'm unsure, because they do look quite different. But because hand pollination was a strategy at the site to increase the numbers back in the 80s, there may have been accidental pollination occurring there. However, with it occurring in 2016, there is the potential that insect-mediated cross-pollination may be occurring. Certainly with Hartslock, purpurea has hybridized with simia at the site in being recorded in 2006. However, the purpurea found at the site appears to be of continental origin based on the DNA compared to local purpurea population. So there is a concern that the purpurea at that site could have been deliberately introduced, and whether or not there was any deliberate cross-pollination, I don't know. However, of course, orchids may be, it's suggested that orchids are very fit for seed dispersal over long distances. Certainly they've managed to colonize a number of oceanic islands, and so natural dispersal can't necessarily be ruled out, but yeah, it is of concern. So we know that there's considerable overlap between all three species in terms of visitors, whether or not they are pollinators. We don't know. Hoverflies seem to be very dominant, and particularly between Anthropomorphic and simia. However, there is phenological overlap with purpurea, but it does vary. There is spatial overlap. Species are occurring in closer proximity at the Faversham site. However, purpurea is currently, except for 2 or 3 plants, behind a kind of thick conifer, kind of wall of conifers, and so it's going to reduce the number of pollinators that can potentially get through and result in cross-pollination potentially. Now, one of the terms that people often use is orchideates. There is a tendency by certain individuals to create hybrids or disperse seeds to other sites. And so this, I think, is a potential concern. So human mediated hybridization, or even if it's not hybridization, introducing plants to other sites is of concern. Now, of course, stitching for 52 hours in front of inflorescences is extremely time consuming, and therefore with funding from the Botanical Society of Britain and Ireland, I've started using camera traps. In this case, the kind of the GoPro-like cameras, because I find it very difficult to find a camera trap that gets triggered by pollinators. And so what I've done is set these cameras going and taking photos every 3 seconds to ensure that we capture anything that goes to the flowers that we're looking at. Now the problem is that I now have 0.25 terabytes of images. Each image is about 5 megabytes. So you can see that's a lot of images to go through. And so currently with the engineering department here, I have students looking at how we can process these images quickly, not necessarily to identify what the pollinator is, but to remove all the images that don't actually have an insect in them. So that, you know, as I say, at best, Orchis simia was having 3 visitors per hour. But if you're taking photos every 3 seconds, that's a lot of images to get through. But you have to take it so frequently because visitation seems to be extremely rare. So how can we process these in a manner that allows us to identify pollinators quickly without all these images? So in terms of final thoughts, of course, there has been a lot of work on the Hartslock site, looking at the work by Richard Bateman, but more recently by others. I can't remember the guy's name who did his PhD recently on anthropomorphic orchis species. But there has been work looking at the genetics and hybridization, and showing that there has been historic hybridization between Orchis simia and Orchis militaris, so the military orchid at the site. So the hybridization is part of natural processes. There are, of course, many species that evolve from hybrids. So it's not as though we should be thinking about preventing hybridization as much. However, I think human-mediated pollination isn't necessarily a good strategy. The species are at the edge of the range, so you're going to get overlap in terms of phenology and potential with pollinator communities that may increase the likelihood of hybridization occurring. And so this brings into question, when you've got such a rare species, 3 sites in the UK, occasional others popping up, but what do you preserve? Do you preserve the species, or do you accept the process in terms of hybridization is occurring and just allow it to occur come what may? Or do you try and do you try and conserve both aspects? If you're going to go for both, which is what I would prefer, is to ensure that the species can persist, but to allow natural processes to occur in terms of hybridization as well, then you have to think at a much larger scale, a landscape scale, allowing the species to be able to move between sites. So you have some sites that hybridization may be occurring at, but other sites where the species where hybridization isn't occurring. Um, you know, now that it's not just in terms of climate change where we think about assisted migration moving species northwards because of climate change, but given the fact that we are in such a fragmented landscape and the likelihood of seed turning up at a site, maybe we need to start thinking about assisted migration, and acknowledge that we are in a very fragmented landscape with large gaps between potential sites, and that we may need to start thinking about moving seeds ourselves. I put this slide in terms of orchid seeds. We have done a little bit of work in the long past looking at orchid seeds, and actually orchid seeds aren't as good at dispersal as we often think. There has been some physical experiments using fans or putting pollinating plants in the wild and putting petri dishes out. But also we've done some kind of theoretical modeling with an aeronautic engineer, and orchid seeds don't seem to disperse over long distances. And it's likely that orchid seeds didn't necessarily evolve for long distance dispersal. Is probably more likely a product of their mycorrhizal association, so the association with the fungus, that there is no endosperm, and therefore that clearly reduces the weight of the seed. And so a byproduct is light seed, but not necessarily designed specifically for long distance dispersal. The final image at the bottom, which is one thing that I was kind of playing around and tried to work out how to draw it, and I kind of gave up at the end, is to think about the environment that we're in. We are in a very changeable environment, and particularly with climate change occurring. And so we have these reserves. We've got the fabric site is a relatively small, only a few hectares in size, which has an important orchid population. But it's like to me, often with conservation, we're trying to maintain the orchid at the site, and orchids can be very transitory. We're trying to maintain it at a site, and it's like being on a seesaw. The environment's moving up and down each side, and we're trying to keep this kind of ball of a species sat in its location. And sometimes the environment will suddenly move and it will fall out of there and it may become extinct. Or if we've got sites nearby, it may land into one of those sites. And this is why I think we need to start thinking about more of a landscape level, and start at least with Orchis simia, looking at other sites nearby that can be restored or developed into sites that are fit for colonization, whether or not it's natural colonization or assisted migration to these sites to allow the species to persist. And with that, I want to acknowledge a number of people and organisations, in particular BSBI and the Botanical Research Fund for funding my research on this. The landowner very much for getting me interested, getting me back into pollination ecology. I'm very, very grateful for their access. Viv Brown for helping me identify hoverflies. Coming into this project, I did decide that I was going to try and get better with my insect taxonomy, and so I went on a number of courses. And so thanks to Viv for helping with those. Mike Watts for a number of the photos, and also the hard work of the volunteers at the site. The species probably wouldn't persist at the site if it wasn't for their hard work. And with that, I shall leave it there.
Fantastic, David. Thank you so much. The Q&A is full, I would say. It's got eight questions in it, one spelling correction. And I don't really know where we start. I think potentially there are just a couple of practical questions about the study. One saying, did you do any studies this is from Sean Cole, sorry. Did you do any studies in 2024? Understand it was a particularly good year for simia at Faversham, so it had been a good opportunity. Um, Perhaps that was covered in
Unfortunately not. I really wanted to, but unfortunately the university was having a change in the um, was changing the structure of our teaching, and I was up until recently in charge of an undergraduate program, so I spent the entire year rewriting all our undergraduate specifications for modules and the degree. So I didn't get to look at any orchids last year, which is rather
And they
depressing.
know, don't they? They know when no one's watching, as we found out. Yeah.
Yes,
Um,
and there are probably loads of pollinators. They're probably going
exactly.
around
That's
flaunting
where
their
they all
pollinia.
were.
Yeah,
Fabulous. Thank you. Joe has asked, and again, I would combine this with James Cartwright's question. Joe asked about the most likely time of day for pollinators. So you said were visits selected at, oh, sorry, were the visitors um, were visits at the same or at randomly scattered times of day? And James said, were they observed overnight to check for moth pollination?
I did check for moth pollination.
Mhm.
Essentially, I was limited by the school run, so I got to the site as soon after 9 o'clock as possible and stayed there until about 3 in the afternoon. Um, so yeah, that's the general time period that I was out there. I tried to stay at the site as long as possible. I
Yeah.
didn't do any night visits. Um, it might be interesting putting moth traps. People have done moth trap work, and nobody's recorded pollinia on any of the moths that they've caught, as far as I know. And given the number of rabbit holes at the site, I potentially wouldn't want to wander around there knowing what I'm like in rabbit holes. Yeah.
Sean makes an interesting point. He has checked the Pollination Bible, Classens and Cleanan, and they list 5 pollinators for Osimia, 3 bee species, 1 moth, and a click beetle, so no
Yes.
hoverflies.
Yeah. So there is a nice paper on from a French researcher, which recorded a number of click beetles pollinating at their site. It's probably most likely that there's going to be, with it being a species with nice flowers but no reward, it's going to be a generalist pollinator, so it'll accept any pollinator that comes along. And it's probably going to be more site specific in terms of the pollinators that an insect diversity that's around at that site.
Mm.
So I know, talking to people at Hartslock, that they notice a lot of bee species on their flowers compared to hoverflies.
Thank you. The rest mainly relate to habitat or establishing new colonies or populations. Marie asked, is it possible to expand populations through plug planting?
Um, I don't know much about orchid reintroduction work. Certainly, Australia and Western Australian scientists are very famous for their work on restoration. Um, so I'd probably refer you to Kingsley Dixon and Kings Park Botanic Gardens for that. Of course, Kew has done some work in terms of um, Cypripedium calceolus and um, Anacamptis laxiflora work as well. So
Amy has a similar question on assisted migration, I guess. What distances from original sites do you feel will be appropriate? How far is too far?
So we've done work on. So if you think about orchids, or the orchid family, and you look at the distribution of species, there's a lot of endemics. When you get to countries like Ecuador, you're talking over 4,000 species. And often occurring in very, very limited locations. And from at least the theoretical work that I've done on aerodynamics and some of the work that others have done, you're talking, you know, distances of maybe only 1 or 2 kilometers in terms of dispersal. And that is per generation. That's not even,
Mm,
you know, yeah, that's based on a generation, not necessarily, you know, every year.
thank you. As an anonymous question who asks, is accepting hybridization different now as areas of orchid habitat are separated due to urbanization, whereas previously they might have been more connected? Has that changed any of the thinking around accepting hybrids?
I don't know. I think it's just a variation in terms of how botanists think about or think about plants in general. Whether or not you're a purist and you know, essentially you have a botanical
Hmm.
museum and you want things to stay the same, or whether or not hybridization is, you accept hybridization is part of this landscape that we live in.
Hmm. Two final ones.
Yeah.
Marie has asked, what are the impacts and benefits on a habitat if orchid numbers increase in an area compared with an increase in a more common species, in the number of more common species?
It I think in terms of, if you're thinking about why should we bother about orchids compared to other common species, I think you know, orchids are, to me, in some ways, the ultimate parasite, in that they parasitize the fungus in terms for germination. They parasitize if they're deceptive, they don't produce a reward, and therefore, in a sense, parasitize the pollinator. And then they parasitize each other, that they try to get rid of their pollinia, get rid of the pollen, and put it onto another individual within the population to maintain that and basically take the cost of fruiting success. So, in a sense, they are very parasitic. They can be very parasitic, but I think that's that is what makes them good indicators in terms of environmental health. And you know, the fact that if you can maintain some of these species at the site, it's a good indication that other species may be able to persist at the site as well.
Thank you. And one last one from Richard Bateman, and I've left this to last because I'm hoping you will understand what he's referring to, because I'm afraid I don't. He said, nice talk. With regard to pollinator specificity, why not also briefly tell the story of the Austrian professor, the beetles, and Orchis italica on Chios? Is that one you know?
I don't know that one.
We might have to ask Richard to host his own talk and tell us that story.
Yes,
I've not been able to find anything in a brief Google, so I'm sorry, Richard, but please do send in, or perhaps when we have the video up on YouTube, you can provide a comment that spells that one out for people. But thank you for that.
I know that I was at a conference. Whether or not he's referring to this, we went to. I was at a conference where Richard was. It was an orchid conference, and we were, you know, with the conference being on orchids, we got to go and look around, and I was finding praying mantis sat on them, and I started wondering whether or not they were whether or not praying mantis could discern whether an orchid has a reward and therefore chooses rewarding orchids because they'll get more pollinators. And so we actually did some work when we came back looking at orchids in the UK, but of course we don't have praying mantis around, so we had to make do with crab spiders and
Mhm.
look at whether or not crab
Mhm.
spiders chose rewarding compared to deceptive orchids. In the end, it was that it made no difference, and the idea. The reason we think is that once a crab spider has eaten a nice big juicy bumblebee, it doesn't have to eat for a while. So it's a bit like a python; it just needs one feed every so
Mhm.
often.
Hmm. Fabulous, thank you. Martha's snuck in with one final one. I think we'll take that as our final one and then wrap up. But she said, pollination success. Have you associated the percentage found with any abiotic factors, e.g. temperature?
No, I haven't. I've been trying to get funding to put a weather station at the site. So a permanent weather station, or at least one that can be up during the season. But unfortunately, I didn't get funding to do that. I did
So
try and plead to the
TBC, maybe some
university, but
future,
yeah,
maybe one for future. Fantastic.
yes.
Well, thank you so much, David. This has been brilliant. I'm sure orchid idiots and orchidophiles alike, and anyone else will be watching it on YouTube and watching any subsequent publications and extra research with interest. But thank you so much. Thank you, everyone who has joined in. As I say, the recording